What are these swirling shapes, this fantastic tessellated mystery? Who made this picture, and how did they make it? Hi, I'm Keenan Mitchell, your host here on Profile of a Scientist. To find out the answers to these questions, we must journey into the Hall-Atwater laboratories at Wesleyan University to meet a painter, photographer, and scientist named Frank Stellabotte.
Hi, I'm Frank. A lot of your training as a young scientist is looking at drawings and looking at pages in books that have sort of sketches of what the nucleus is supposed to look like and what a cell is supposed to look like and what a cell membrane is supposed to look like and what different structures of one's anatomy should look like. But when you look into an electron microscope, you can see it, you come face to face with every individual component of the cell. And that was pretty amazing to me, actually, because here I'm seeing it for the first time in real life. It's sort of like running into a famous person on the street. So that was a very important part of my development, I think.
So he began taking pictures of what he saw through the electron microscope. These pictures, when colored, yielded works of art like these, and the image we saw earlier.
And it was then that sort of the art and the science came together. The electron microscope I was using is very old. It did have a digital camera, but it wasn't very high resolution, so all of my images were taken on film, using a film camera. So of course I had to develop all the negatives from the microscope, and then I had to make prints out of all of my images that I took. And so it was very easy for me to apply my techniques that I learned in the art studio to the research questions that I was trying to answer there.
The research he's referring to is his dissertation work. It is for this that Frank began studying muscle development in zebrafish.
And what I did was I looked at the embryo and I picked out individual cells, and I looked at the fate of each cell. And then from there I was able to determine which cells gave rise to muscle right away, which the fish uses to swim away. And then I discovered which cells give rise to growth of that initial muscle population, just by studying one cell at a time in the embryo.
But how does all of this relate to his artwork? How are the goals of his scientific research and his personal artwork similar?
In my paintings, movement and motion is very important to the composition. And in my scientific research, one of the most important themes are the movements of particular cell types, and conveying that sense of movement from a static image is very important to my research. So those techniques I learned in my paintings, and it's really interesting that the sort of the artwork has significantly impacted my scientific research.
But why was he studying zebrafish in the first place? Who could tell us why they're such a great model for studying muscle development?
Oh, definitely a good person to ask would be Betsy.
There's actually a large number of reasons. One thing that's really good about zebrafish is they're vertebrates, so they're closer to humans than some other models, so we can apply what we know about zebrafish to humans. But what makes them particularly good are a number of things. If you want to study development and you want to watch an embryo develop, it's really nice to have something that's transparent. So the zebrafish are clear for quite a number of days. You can see the heartbeat, you can watch the blood flow, you can watch cells, where they go, so you can photograph them through time and get a good idea of what's happening and watch cellular behavior. And they develop outside of the mother. So the males and females will have a mating behavior, and when the lights come on in the morning, they'll lay eggs. And then you can collect those out of the water, and you can put them on a microscope and watch them develop.
And, you know, as a species, they're pretty good looking too.
His appreciation of nature's beauty and an eye for aesthetics has guided Frank's work in the lab. But don't take my word for it. Let's hear it from him.
If I wasn't so interested in art, I would never have completed the project that I worked on in the laboratory. The reason I decided to study the cell population was from constantly looking at individual cells, and not so much with a scientific purpose in mind, just because the sheer beauty of the embryo at that stage and just the amazing structures that you can see and different cells that you can make out just fascinated me so much that I stared at them for weeks before deciding that I wanted to study a particular cell population. And much of that sort of appreciation for the embryo and for the individual cells and for sitting at a microscope for hours looking at different cells came from interactions with my advisor, Stephen Devoto, and he really taught me that.
So many of our results come out as visual results rather than, for example, as graphs or numbers or tables. We're looking very directly at the muscles themselves in the microscope. And the collecting those results and analyzing them requires a lot of time looking at what I think are quite beautiful stained zebrafish embryos.
But to Frank, they are not just beautiful pictures. He takes it a step further, erasing the distinction between science and art.
But my sort of desire to sit there and look for hours and hours at cells and seeing how some of them are different than each other, and that came out of my sort of training as an artist, just to appreciate the beauty of the image and of what's going on there.
So to me, there is no distinction between science and art.